I'm sitting in here or standing in for Richard Showman, who is under the weather today. And I'd like to introduce Jesse Jokerst, who is a professor in the Department of Nanoengineering at UC San Diego here. And he graduated from his PhD is from chemistry at the University of Texas in Austin. And he did a postdoc at my university, Stanford, in radiology from 2009 to 2013. He's now at the University of
Your sound mic is off.
Hold your mic closer.
Okay, yeah, I can talk louder too. That may be the other answer. Okay, and he's actually he's now principal investigator of multiple federal grants and serves on the editorial advisory board of ACS Applied Nanomaterials and Nanoscale. And the university here has a pretty strong program in nanoengineering, and that's what Jesse's involved in. So without further ado, here's Jesse Jokerst.
Thank you, Ralph. Thanks for filling in at the last moment, and thanks for the invitation. Hopefully my audio's okay. So yeah, it's great to be here. We'll
just put this down here. Make sure it doesn't roll off.
Sure.
There we go.
So the way my lab is kind of set up right now, and the way I'm going to organize this talk is kind of along this triangle. And so on one corner of the triangle down here, I'm trained as a chemist, and so about 50% of the work in the lab right now is chemistry and material science. So we're making new materials, we're making new molecules that respond to disease or respond to human health. There's a corner down here which I'll call real engineering. So this is, I have students who are electrical engineers, optical engineers, and they're making new devices that detect sound or new software that process images. And then there's a corner up here which is human subjects research. And so I have 4 active human subjects trials. And so these are in oral health, wound care, COVID-19, and therapeutic drug monitoring related to heparin. And so these parts sometimes don't fit together very well. Sometimes they can kind of stand on their own. So the way I organize this talk is to just kind of tell you a few stories. And so these stories aren't always going to connect to each other very well, but they will always come back to this triangle. And so part of the glue that holds my lab together is imaging, and particularly ultrasound imaging. And so if we look at this figure, what I'm showing here on the x-axis is the temporal resolution. So how fast can we collect an image, right? And so many of you have probably had an MRI or a CT, and you know when you're put in the MRI or the CT, it takes a long time, right? It's like taking a photograph. If you think about ultrasound's like taking a movie, right? You have the scanner, it's very fast, we get video frame rate. So ultrasound is exciting from that perspective. It's also exciting when we look at the y-axis, which is the spatial resolution. So this is what is the smallest feature size that I can resolve, right? How blurry is my image? And so ultrasound is not as good as, say, microscopy like we would get in optical imaging, but it still has really good resolution relative to things like PET and MRI. And so depending on the audience I'm talking to, people tend to think of ultrasound as something that's used in pregnancy, and it's very powerful in obstetrics, but it's also very useful in cardiovascular medicine, abdominal medicine, reproductive medicine. It's also by far the most affordable and universal piece of imaging equipment everywhere from UCSD Hospital, Scripps Hospital, to a private practice clinic. They all have ultrasound. And so these are some of the features that got me excited about working in ultrasound. So some of the problems in the field is that it still has relatively low contrast. And so what do I mean by contrast? Contrast is just the difference between the area we're imaging and the background, right? And if you want to have that image pop, you want a big difference between the signal and the background, right? So this is an example of an ovarian cancer mass next to the urinary bladder. And so if you're a skilled radiologist, you can pick this out as being an ovarian cancer mass, but it's pretty difficult. And this is an image that has not a lot of contrast. And so when I moved to San Diego and started my lab, one of the first things we did is look at a variety of materials that we could use to specifically increase the ultrasound contrast of images. And so here are just some of those papers from the first few years of the lab. This was led largely by Fong Chen, who was a PhD student, is now at Stanford, and Tae-Ho Kim, who's now a professor at the University of Michigan. And so these are just some of the materials. I obviously can't tell you about them all, but what kind of holds them together is that they are on this nano scale. And so what exactly do we mean by the nano scale? For those of you who have been following the new James Webb Space Telescope, you've been seeing a lot about dimensions that are really, really large, right? Like light years away. This is dimensions that are very, very small. And so if we think about scaling down by a factor of 10, this is a logarithmic scale, from a tennis ball to a period to a cell to a bacteria to a virus, antibody, glucose, and then a single water molecule. So it turns out that the field of chemistry is really good at controlling matter on the very small size regime, right? Making molecules, making maybe slightly larger molecules like a polymer. And then, humans are pretty good at making things with lithography or printing or other things in this regime. But this area here between, roughly 1 and 100 nanometers or 10 to the negative 9th meters has really within the last 20 or 30 years been an area of tremendous growth and tremendous opportunity. And part of the reason this is important is that material matter behaves differently when it's on this light scale. So I'll show you an example later of gold, and I can make this gold look blue or I can make it look red, right? And normally you think, you all know what the color of gold is, right? But by changing the size of it, you can change the color of it. You can change how it interacts with matter. And so that's just one of many examples of how matter behaves differently when we put it on this nanoscale. So what does this have to do with ultrasound? Well, one of the first applications that we looked at was in cardiovascular medicine. And so there have now been about 10,000 people who have had cardiac stem cell therapy funded in large part by the California Institute of Regenerative Medicine, or CIRM. And so in these cases, people have taken stem cells. Generally it's from the bone marrow of the patient themselves, and then these stem cells from the bone marrow are grown in a lab, they're expanded, and then they're injected into the heart of the patient, generally, frequently under ultrasound guidance. So the problem, though, is that these cells are being put into a sick heart, right? This tissue is inherently necrotic, hypoxic, meaning it's sick, right? And so to take these cells and put them into a really difficult area of the heart means that a lot of them die. And so what we are using are these nanoparticles, so things that are much, much smaller than a cell, and they have ultrasound signal. They can also serve as a reservoir of a drug. So we can put a drug like insulin-like growth factor, which is basically a vitamin, and we can put this vitamin inside the cell. So this helps the cell stay alive longer. And we can also use the imaging to understand how many cells are there. Are the cells alive? Are the cells dead? Are they interacting with the surrounding tissue? And so this is what we would describe as a multimodal particle. It's doing multiple activities at the same time. So this is an example of what you're looking at here, let me tell you. So this is a mouse. This is a mouse that's laying on its back. And then I have an ultrasound transducer going this direction. So you're looking at a plane into this mouse's chest. And so when I click play, you'll see the beating of the left ventricle. LV is left ventricle. And then this line right here is a catheter that I've inserted into this animal's chest cavity. And this region right here is the region in red is where I'm going to deliver the cells. So you can see relatively low signal, low signal, low signal, and then now you can see these cells that have been injected into the left ventricle wall. And so without these nanoparticles, we would not have seen these cells. But because we have this contrast agent, I know that this therapeutic event was successful, that the cells got to the region of interest. And then we could also, because of the way that we have built these materials, we doped in gadolinium. Gadolinium has more unpaired electrons than anything else on the periodic table, so it's a really good MRI contrast agent. And so now in this same animal, I can look pre and post-injection, and I can see this white signal right here, which indeed, again, corresponds to the left ventricle and the location of the stem cells. So we wanted to understand a little bit more about the efficacy of these cells. And so what I'm showing you now are two heart images. And so on the left is a healthy heart, on the right is a diseased heart. And when I hit play, you'll see these hearts pumping. And so what we're now looking at is what's known as a long axis view. So I'm looking at this plane right here. And so this is the tip of the heart, or the apex. And this is the aorta right here. So you can see this heart is ejecting, blood is going out of the aorta. You can see the atria right there, and there's the left ventricle, right? And so on the left, this is a healthy heart. You can see how thick the left ventricle wall is. You can see how it's ejecting most of the blood that's in the chamber. On the right, this is a sick heart. This is an animal that we've given a heart attack to, right? So you can appreciate, look how there's very little movement right here. Look how thin the left ventricle wall is. This heart doesn't have a lot of muscle. And look at what's happening to the blood. It's kind of circulating back on itself, right? There's not a lot that's getting ejected out of the aorta. So this is what's known as the ejection fraction, right? What fraction of blood that's in the chamber is actually being ejected? And so we can use ultrasound to calculate that metric called the ejection fraction. What fraction is getting kicked out? And so through a lot of different studies, we looked at a group like this, where group 1 is healthy animals with no heart attack. And so they're ejecting about 50% of the blood, which is normal for a healthy mouse. In our area, group 2 had a heart attack but no treatment. And so they're reduced down to about 30%. This group 3 was given stem cells, but without any therapeutic nanoparticles, and so these cells really quickly died because, like I said, they're being put in this hypoxic and necrotic region of the heart. But this group 4 was given cells with nanoparticles, and so we showed through other mechanisms that these cells are staying alive and proliferating, and indeed we're getting this improvement in how much the heart ejects when we look at this over time. So by 60 days, we're seeing this significant increase in how much blood is ejected from the heart. Okay. I think I'm going to skip this next story because it's too applied or basic. And I'm going to talk to you about a totally different project that again relates back to these two axes. So again, I'm going to use chemistry and contrast media, and I'm going to try to use that to improve human health. And this is a topic that we're all familiar with, is wearing these face masks. And so this is a project that is in response to a call that NIH released, the National Institutes of Health released, related to surveillance techniques. And so this was money related to the RADx program, if any of you have heard of RADx. And what NIH wanted here was a tool that did surveillance. And so what do we mean by surveillance as opposed to detection? Surveillance is what a smoke alarm does, right? It's constantly working, it's sitting in the background, and when a smoke alarm goes off, it doesn't necessarily tell you there's an electrical fire in the bedroom or a grease fire in the kitchen. It just says you need to take action, right? And so that's what this call that the federal government had was for tools for doing surveillance of COVID-19. And so at the time, face masks were completely ubiquitous. And so we thought, what better way of doing surveillance than maybe turning the face mask itself into a tool to monitor disease? And so the vision is a sticker that can go on any kind of conventional face mask. And so you would pull a backing off and stick it and wear it at the beginning of the day, the beginning of a shift, the beginning of an airplane flight, maybe the beginning of a lecture. And at the end of it, you would click this little blister pack that would release a liquid, and it would flow on this little absorbent pad. This is the same pad that's in a home pregnancy test. And you would look for a color change. And if it's both blue or kind of more purple, this indicates that you might have been exposed to a SARS virus. If one is purple and one is pink, then you're negative, and if they're both pink, that means that it's a bad test and you should throw it away. And so this is led by Ziqing Zhen in the group, who's a very talented postdoc trained in Florida and is going on the faculty market soon. So what are we testing for? I'm sure you're all quite familiar with the different kinds of COVID tests, right? So there's the polymerase chain reaction, right, which looks for nucleic acids. There's a rapid antigen test, which looks for antibodies. We're looking in this case for what's known as a protease. And so this is really complicated. You don't need to really pay attention to this, except to know that a protease is a protein that eats other proteins, right? It goes and it finds a specific section of a protein and cuts it in two, okay? Why would a protein want to do that? Well, proteins want to do that in order to make proteins in the right form, right? And so what SARS viruses do is they have these proteins, and I'm going to call this one the main protease, or MPRO. It's the main protease, and it's involved in the viral life cycle. So this is a good biomarker for COVID-19 because this protease is inherent to the viral life cycle. Paxlovid, this is the drug that's been approved to treat COVID-19. This is the target of the drug, is this main protease. There's no close homologs in humans. It's also well conserved. So this protease was present in the original SARS in 20 years ago, SARS-CoV-2, MERS, Middle East Respiratory Syndrome, and it stands to reason that SARS-CoV-3, should there be SARS-CoV-3, will probably also have MPRO. The other thing that's great about a protease is it's kind of like a catalyst, right? And so with a PCR, you inherently amplify the signal by using different thermal cycles. And here I'm just going to use the protease itself to kind of catalyze and amplify the signal. Okay, so that's what we're trying to measure. This is how we're trying to measure it on this mask. And so how are we getting this color change? We're getting this color change, and this is part of the magic of nano, is that we're taking these little bitty particles of gold. And so that's what I'm showing you right here, is this is a transmission electron microscope image. And so you can see these little pieces of gold. And believe it or not, when they look like this, they're red. And when they're stuck together like this, they're purple. And it's pretty complicated how that happens, but basically you're getting the differences in how this interacts with light, and you're getting differences in scattering. And so this both absorbs and scatters light differently than a small particle. And so how can we make this change from red to purple or pink to purple or pink to blue? How can we make that specific for this biomarker? Well, the way we do it is we build this peptide sequence right here, and a peptide is just like a small protein, and these are different amino acids. And so this sequence right here, TSAVLQ, is what the main protease knows to look for, and it cleaves right there. So normally this protein, this peptide, has a positive section and a negative section. And when they're bound together, they balance each other out, right? But when we cleave right here, when we cut it with this protein that eats another protein, this positive fragment aggregates these particles and causes them to collapse on top of each other. And so what does that look like? Well, here it is in a tube. And so you can see this color from red to bluish. And here you can look at the rate at which these colors change. And so we're detecting within about 10 or 20 minutes at a concentration of 20 to 30 nanomolar, which is reasonable for diagnosis. We then looked at how does this color change in different media, and so we looked at buffer, and obviously behaves very well in buffer. We looked at saliva. It behaves actually pretty poorly in saliva. Saliva is a very difficult matrix. It's thick, it's very mucousy. But what we show is good behavior in exhaled breath condensate. And exhaled breath condensate is just what it sounds like. You take your breath, you breathe over something cold, and it collects, right? And so for a virus that lives in your lungs, exhaled breath condensate is a pretty good diagnostic medium. So I've learned a ton about exhaled breath condensate, EBC, in the last couple years, and have been using it for diagnosis because this is where this little sticker would live. So this is, we've built this sticker like this, we've put the particles here, and so this was a study where we took healthy exhaled breath condensate and pipetted it on the sticker. And if you trained your eye left versus right, they're different, they're different, they're different, they're different, they're different, they're different until we get about here, and then above 40 nanometers, both lanes are the same color. So this is our detection limit when we're on this little absorbent sticker. We also wanted it to be have no false positives. So here we had 10 people who were PCR negative, wear this sticker over the course of an 8-hour day, and then click the little blister pack and look for a color change, and we had no false positives. So I wish I could talk about this in more detail in terms of our clinical work. We've now looked at 50 positive undergraduates at UCSD and 50 positive people at the VA. And what I can tell you is if I was to redesign this study, I wish I would have done it longitudinally and imaged or tested the same people over time, because it turns out there's kind of a dynamics in how this protease is expressed over time. But we are detecting a large number of these positive subjects, so that's encouraging. Maybe I'll go through this quickly, but just to tell you about a project that's led by Maurice Rateau in the group. He's a Belgian postdoc, and he's making these even more remarkable nanostructured materials. And so these almost look like snowflakes, right? This is called diffusion limited aggregation, and it's kind of like what you imagine a black hole doing, where it sucks in these planets and pulls them in and puts them in a combined dense system, because you can see these particles being pulled out of solution and grown into this higher ordered structure. The reason we're interested in this is because the difference between pink and purple can be pretty subtle, right? But the difference between yellow and blue is much more pronounced. And so we're using this color changing approach to try to make the signal difference more obvious. And so what Maurice has done is again tried to link this back to how MPRO can cleave this protein and cause this growth into a silver-like structure. And so again, we've shown this and show that we have good detection limits. We can detect this by eye, we can detect it on a plate reader, we can detect it within about 45 minutes. And then more importantly, we can discriminate between SARS-CoV-2 and influenza. So influenza right here is in green, SARS is right here. And the way we're doing this is using a library of peptides and looking for multiple kinds of color changes. And so obviously going into the fall, this is an important question, is flu versus COVID. And so if you could have a colorimetric way of detecting this at home without having to visit a physician's office, this could potentially have significant value. Okay, so going back to the big triangle then, I want to talk about a couple projects on this axis. So here we really don't have any nanoparticles, we don't have a lot of new chemistry, but we are impacting patients. And so the first one I'll tell you a little bit about is in wound care. And so this is a collaboration with Pranav Garamella and Cesar Anderson, both at UCSD. And so you probably know people that have these diabetic foot ulcers or renal insufficiency injuries, these kinds of wounds that develop and just don't heal, right? This is a real problem. So Caesar sees these people day in and day out, and it has a major negative quality of life impact. And so really what they do, they go in, they can it's called debridement, where they basically scrape it out. They can do a skin graft. They do what's known hyperbaric treatment. So they roll people into this chamber and seal the door and turn it up to 3 times normal atmosphere and have them breathe oxygen to kind of force oxygen into the wounds. But there's really no way of knowing, is it working or not, right? And so what happens is that they just keep coming back week after week and trying something different. And so we thought there has to be an easier way. And so linking this back to ultrasound, what we're using here is this technique called photoacoustic ultrasound. And so what this is in conventional ultrasound, like I showed you with the heart, what we were doing is sound in, sound out, right? We had some sort of sound producing device. It's emitting sound pressure waves that are bounced back and detected. What we're doing here is light in and sound out. And so this was originally described by Alexander Graham Bell over 100 years ago, but it hasn't been until the last 10 or 20 years that people have been using it for imaging. So how does it work? Well, it turns out that as these photons are going through tissue, they're absorbed and they can be scattered. And normally in a lot of optical techniques, if the photons are scattered, it really basically ruins the image. But in photoacoustics, I don't care how many times they've been scattered, I only care about when they're absorbed. And when they're absorbed, there's going to be a very short thermal expansion event, right? So you know that that's why hot air balloons rise, right? It's thermal expansion. And so we're doing the same thing here, but because I'm shining this light in a very short amount of time, there's no temperature increase, right? There's no time for the heat to propagate. And so the heat is it's spatially confined. And that causes this tissue to basically wiggle, right? So I'm causing this tissue to wiggle based on incident light. Okay, and so as these pressure waves are back propagated, they're detected by these transducers, which is just a thing that detects ultrasound, and then I can reconstruct my image and it looks like what was doing the absorbing. Okay, so that's pretty cool. What's also cool about it is that I can do multiple wavelengths. So I can excite with red, green, and blue ultrasound, or excuse me, red, green, and blue light, and I can get different channels of ultrasound information, which is generally quite difficult to do with conventional ultrasound. So I still have good contrast, I have good resolution, I can look at multiple color channels of information. Some of the limitations of this technique, I still have to get light through tissue, so like getting through adult human skull is going to be hard. It's hard to get photons through that. It's hard to go deep into the body. But there's a lot of really interesting biology in the first 3 to 5 centimeters of tissue. Okay? So what is doing this? What's absorbing the light? Well, it turns out that hemoglobin and deoxyhemoglobin have really strong and really different absorption spectra. So here I'm showing how does this absorption change as a function of wavelength. So this is more blue light and this is more red light. And so what I can do is I can do 2 different colors of absorbance. And so if I have the known molar extinction coefficients, then I have 2 equations and 2 unknowns, and I can back propagate not only how much total hemoglobin do I have, but what fraction of it is oxygenated. So that starts to become pretty powerful, right? It's not entirely unlike how a pulse oximeter works, right? You wear the pulse oximeter, it tells you how oxygenated you are. But what this is going to do is make a map. And I can make a map in 3 dimensions anywhere I put this transducer, right? So if we think about diseases like these wounds that are very hypoxic and necrotic, this starts to become a very important way to monitor a response to therapy, maybe predict the emergence of wounds, and even predict the best therapy for a kind of wound type. So I'm not going to spend much time on this, but except to tell you that the engineering component here, there was a ton of work in the back about optimizing this light source. So when this technology first came out, people were using these big bulky lasers, and recently my group has been using these LEDs, and this was read by Ali Hariri and his colleague Moeen. And so when you think about an LED, it's not entirely unlike a laser pointer. You can drop it on the floor, you can throw it in the back seat of your car, you can move it around the hospital, bump it on a cart, no problem. Try doing that with a conventional laser, it starts to become difficult. So all of these clinical studies that I'm going to show you was done with this LED excitation and using LEDs to have these really short light pulses. So the actual scanning was done by Yash Mantri, who recently graduated and went to work for Illumina. And so what we did were image these human subjects over time. And so every time they came into the wound clinic, Dr. Anderson or his nurse took off the bandage, we imaged them, and then bandaged them up again. So really simple, really fast, and we could monitor these patients over time. So here's an example. I like this example. Here's a photograph of the wound. This is kind of on the back of the leg. Here's the first day of treatment. And so the gray image is regular ultrasound, sound in, sound out. The colored pixels here are the photoacoustic data. And this is just total hemoglobin. How much blood am I getting to the wound site? And so you can see at day 1, there's not a lot of perfusion. You can also see this black area right here. I'll draw it in if you don't believe me. But so this is this wound area, this very hyperechoic region. On day 29, you can see that the region of the actual wound is significantly smaller, and I have more hemoglobin signal coming into the wound. I also have more scar tissue down here, right? We can see a little scar tissue here, a lot more scar tissue there, that brighter signal. So these are really important biomarkers of understanding this response to therapy. How else are you going to do this? You're going to look at the wound and say, is it getting better, right? The other thing I love about this image is that the plane that I'm actually imaging is this dashed line. So Dr. Anderson, when he looked at it, didn't realize that the wound actually extends multiple centimeters away from the opening. This is called a tunneling wound. And so the fact that we can identify this tunneling, which by visual inspection would be impossible. This scan took 5 minutes. The equipment cost $50,000. Compare that to MRI, CT. This is a win-win. We can also look at, I showed you this rate of change. So here's another patient responding. This is a wound on the left ankle. And so this patient is responding, and you can see this increased blood flow right here, this angiogenesis on top of the fibula. Compare that to a non-healing wound. Here's day 1. 90 days later, this patient is still coming back. This still has this wound that is not responding and has really very little difference, in fact, even less blood flow than she did when she first presented. So what we can do is look at that rate of change, but over time. So a healing wound over time is going to have more photoacoustic signal corresponding to more total hemoglobin. A non-healing wound will be flat. And what we can do is then stratify those slopes, and if we're plotting here the rate of change over time and look at how long was the patient in the clinic, we see this bin right here and this bin right here indicating responders and non-responders. And we're really excited about this because this was the slope within the first 30 days, and 30 days is when Medicare and Medicaid reevaluates whether they're going to continue covering a type of treatment. And so if we can convince Center for Medicaid Services that they should continue or discontinue a service, it's good for them and it's good for the patient, right? Because you don't want a treatment that is not working. And so this is a really active project. I mentioned hyperbarrier. So that's when they put the patient in this chamber and turn up the volume or turn up the atmosphere. This is the same kind of treatment that scuba divers get. And so this is a real hassle. People do this for 2 hours a day, every day for 2 weeks. And so if I could tell you within the first 3 days you're responding or not responding to this treatment, that would have a lot of value. And so what we're doing now is looking at oxygenation. So this is healthy control subjects pre and post hyperbaric oxygen treatment. And so this is just imaging of the left forearm. And so pre and post, and you'll notice this increased red signal. So now the color pixels, this photoacoustic data, it has red and blue character corresponding to oxygenated or deoxygenated states. So more red is more oxygenated. And we're very consistently seeing this increased red signal after hyperbaric oxygen treatment, which makes sense. We've forced more oxygen into this patient's body. So we got to do one wound patient. It's in review. The images actually look beautiful. And then Yash graduated, which is always my problem running an academic lab, right? You get the person perfectly trained and then they leave. Start over with somebody new. But this study is going to continue. I think in the flyer that I sent over, I mentioned dentistry, and that's actually another huge project in the lab. We have a lot of work in oral health, and it's also on this axis right here. So the inspiration for this was the last time that I actually went to well, not the last time, but one time that I went to the dentist, and they poked this little metal stick in between my tooth and my gum. And they said 3, 3, 4, 5, and then they scolded me, right? That's like the first lesson in dentistry, I think, is scold your patients. And so what they're doing when they poke that little stick is they're looking at how well attached is the tooth to the gum. So if this is your tooth and this is your gum, they should be stuck together pretty well, right? So when you poke that little stick down in there, it should not go very far. And so the numbers that they're measuring is how many millimeters can they poke that stick down, right? If your teeth are getting ready to fall out, it can go down really far, right? And so that's when you get 5, 6, 7 millimeters of pocket depth. And so I thought there has to be a better way. And working with Coleman Moore, who was an NSF fellow in the group, we've been working on developing an ultrasound mouthpiece that you would just basically bite down on, and it would go zip and quantitate those probing depths automatically and beam it back to the chart. And so ultrasound is ideal for this, right? Because how are teeth currently imaged? They're imaged with x-rays, right? And x-rays are great for hard tissue, bone, dent, and enamel. But a lot of the disease is in this soft tissue, and that's what ultrasound is really great at imaging. And so some of the first studies we did on this are now almost 5 years old, and these were with swine models. And so here again, the black and white images is regular ultrasound. The color pixels are photoacoustics. And so because I'm doing spectral ultrasound, I can look at different kinds of absorbers in the mouth. So these pigs did not take very good care of their teeth, and so they had a lot of stain on the teeth. And so that's what this blue is right here. Here's the gum line, or the gingival margin. And then after we use this oral rinse, we can really easily see the periodontal pocket, what would be probed. And we can measure how deep this periodontal pocket is. And so what I'm going to show you now on the next slide is a slide going into the board. And so that's what these are here. So this is the top of the tooth. There's the root of the tooth, and there is the gingiva. And so this red data right here is the periodontal pocket. It's how deep is that stick going to go. And so I can look at shallow, intermediate, or deep pockets, and then we could correlate it to the conventional probing depth and show that high is high and low is low. So we've done this now in a number of human cases. There's no dental school at USC, at UC San Diego, so we're working with Casey Chen at the University of Southern California. We're also looking at a lot of, I've learned so much about dentistry in the last few years, but a lot of these different different parts of the mouth anatomy. One that's really important is called the cemento-enamel junction, and it's where you have bone versus bone with dentin. And if your gum recedes past that cemento-enamel junction, you have bare bone exposed, and then bacteria eat it, basically. So you want to have all of your bone covered with dentin and enamel. So what we're showing is, can we use this high frequency ultrasound, so this is about 40 megahertz ultrasound, and start to identify some of these oral biomarkers, including the cemento-enamel junction and how much gingival recession you have. So in this study, we included 10 healthy subjects and imaged 34 teeth, and 6 diseased subjects, and we imaged 32 teeth in those subjects. And we did conventional physical probing and oral workup, and then also acoustic imaging to look at these different biomarkers of disease. And what we showed is that for both clinical attachment loss and the alveolar bone loss, we can discriminate between this just as well with our imaging-based approach as with conventional analysis. And so our bias here is less than 0.25 millimeters. So this is suggesting that this is just as good as the conventional probing, but completely non-invasive, right? All I'm doing is just setting this ultrasound transducer on the gum line, okay? One of the biggest problems with this is that ultrasound transducers tend to be big, right? So we can only image the teeth that are in the front of the mouth, the canines, the incisors. And so we're engineers, and so there's a lot of effort in the group to make these smaller, more compact ultrasound transducers. And then I've been fortunate enough to one of my former students, Ali Hariri, and his friend Reza Nazad, we've founded Stylosonics, which is a small startup. So we only have 2 employees right now, but we've actually raised enough money that we've been turning down venture money because we just don't want anyone else telling us what to do. And so have had a few SBIRs from the federal government to support this. And so this is ongoing. And the product is this smaller ultrasound device that can access the entire mouth. So maybe just the last story I'll tell you is kind of combines all 3 of these elements and is related to heparin. Heparin's a blood thinner. Some of you may be on Coumadin, and Coumadin's a blood thinner. Heparin is given intravenously. It's given in the hospital. It's people undergoing surgery, people with blood clots, deep vein thrombosis. It's a really good blood thinner. It's a really good anticoagulant, but it's difficult to manage. And so what they do is periodically do a blood test to try to titrate the drug activity into this region that's both safe and effective. So if you have too much, you're going to bleed out. If you have not enough, you're going to clot. And so how can we get the drug activity into this window really quickly? Well, the convention is to draw a blood sample, but what if we could do imaging? And instead of every 12 to 24 hours, what if we could make a measurement every 3 minutes and really quickly titrate the patient into that window where it's safe and effective? And so that's the vision here, is that we would have some kind of wearable sensor that would be feeding back to the IV infusion pump and telling the pump, give more, give less, this is how much drug is needed. So this is again using this photoacoustic technique, and part of the magic is this electrostatic interaction between this very simple FDA-approved drug, methylene blue. It has a delocalized positive charge, and the backbone of heparin. So heparin's a polysulfated glycosaminoglycan. And so when that positive charge interacts with the negative polysulfated backbone, you get dimerization. And what basically happens is the fluorescence goes down and the photoacoustics goes up. So more of this absorbed energy is released as a photoacoustic signal. So these are capillary tubes with the same amount of this molecule, but increasing amounts of heparin. And I get this really obvious increase in my photoacoustic signal. So what do you do with that? Well, our first approach is to just coat it on the exterior of this intravenous catheter. So this is not an at-home test, right? This is for people who are really sick and probably already have 2 or 3 catheters. And so you would have I'm showing here a big ultrasound transducer, but there's lots of smaller ultrasound transducers that would just go over the top of this and then would monitor the color change or the signal change of that catheter that has the drug-sensitive layer. So we've built a few prototypes like this. This is work with Bill Penny at the UCSD-VA, where we've taken this catheter and tested it both with mock samples and also real human samples. So we're not put this in a patient yet, but what we have done is take blood samples from Dr. Penny's patients in the cath lab, measure it with the gold standard approach, and also dip our catheter in that blood sample and then image that. It's the difference between that and putting a device in patients is significant. So this is data supporting that next step. But what we're showing is this really nice correlation between our photoacoustic signal and the gold standard, which here is this activated clotting time, which Dr. Penny is measuring in the operating room. So moving forward there is we're working on larger animal models. So to put the catheter in this rabbit ear, for example, and then also moving away from those kind of handheld transducers that I showed to more flexible, simpler photoacoustic transducers. So, with that, where is this field going? I'll tell you a couple things that I'm interested in. One, if we're putting light through people, one big variable is skin tone, right? And so if you have a patient with a dark skin tone, they have more melanin in their skin. As the photons try to go through it, there's going to be more absorption and scatter and less light that interacts with the underlying tissue, right? If I have white skin, there's not as much melanin, more light can get in. And this paper got a lot of attention in the New England Journal a few years ago, where they looked at white patients and black patients, and they looked at what is the average oxygen saturation measured with a pulse oximeter. And what they're showing is that black patients were more likely to be misdiagnosed with low oxygen saturation, and therefore less likely to be given supplemental oxygen. This was very controversial, but a paper just came out this week in the Journal of the American Medical Association that kind of reconfirmed these studies, okay? Here I'm showing why, right? If we look at the absorption of hemoglobin and deoxyhemoglobin, I showed you this earlier, here's the absorption of melanin, right? The absorption of melanin is going to compete with that. So we went and looked at how does there's lots of different technologies that try to put light through tissue. Photoacoustics is not the only one of them. But when we have this really dark subject, so what's known as a Fitzpatrick skin type, Fitzpatrick type 6 versus Fitzpatrick type 1, look at this Fitzpatrick type 1. There's very little surface absorption. We can see these blood vessels underneath the skin surface. And if we make a 3D map of that region, we can image this subject's vasculature very well. On this black patient with very dark skin tone, there's so much absorption on the skin surface that a lot of the blood vessels, we can see them with conventional ultrasound, but we can't see them with photoacoustics, and therefore we couldn't measure the oxygenation. And when we make this 3D map, it's kind of really blurred by the high surface absorption. So we've been working on software ways of solving this, and also Wanjun has been working on developing new phantoms and improved. Phantom is basically just a fake piece of tissue. So we're working on building these fake pieces of tissue that we can use to train our algorithms to solve this problem and to learn ways of solving this problem. This is an engineering problem. So this should be a solvable problem. Okay, so with that, I want to thank my group. I feel super lucky to have, every day I get to go work with really bright, ambitious, hardworking people from all over the world. I also want to thank the federal government through the NIH, the NSF. There's my email. Of course, I wouldn't be doing my job to not remind a captive, affluent audience that all donations to the University of California are tax deductible. And thank you again for the invitation and your attention. Yeah, sure. Any questions?
Get the microphone.
Or I can repeat the question.
In many lectures that we have heard, there is a period for trying it out before mass people get it. Do you have that problem, or because it's engineered, it goes, if it works, it works?
No, nothing. So the question is, in terms of translating it from the lab to broader human work, where are you? And in all of them, we've done this in about 100 subjects, right? So something like putting a catheter into people is going to be way harder than imaging their teeth, right? And so, FDA regulatory guidance is always a function of safety and efficacy. And so the safety concerns about a new device is significantly lower than a different way of imaging the wounds. And so, none of these, nothing I've shown you today is commercially available yet, but there's progress in all of them.
How fast?
How fast, it all comes down to money. I think the first one will be this ultrasound one, and I mean, we have a really great working prototype, so I'm hoping that this will be on the market in 3 to 5 years. Yeah.
Anybody else?
Sure.
This isn't working, so. We'll
Just a question. When you talked about the heart and the nanoparticles, you showed that when you put nanoparticles in there, the cells survive longer. But you didn't explain why. What do the nanoparticles do?
Sure, yeah, so the nanoparticles kind of act like a sponge that hold these pro-survival agents. They're basically growth factors or vitamins. And so because the cells are going inside of a very necrotic region, the cells use this growth factor and can, they survive basically on those growth factors. So that's what keeps them from dying so fast.
Nanoparticles are made out of what?
They're made out of silica, and so silica is a common ingredient in foodstuffs, and it degrades into silicic acid and is excreted renally. Yep.
Anyone else?
Sure. Yeah.
When those stem cells are put in the heart, you say they're surviving on the particles, but you want them to integrate with the tissue of the heart.
Yeah.
Right?
Sure, yeah. It's not as if we're getting 100% cell survival, but if we can increase survival by 20-30%, that can make a difference in how many integrate into the heart. And it all, I mean, so this is the engraftment of the cells is generally, this is not FDA approved either, the stem cell therapy. But what most trials have done is deliver this shortly after a heart attack. And so shortly after a heart attack, that's when the heart is most ischemic. So if you can have the cells be there for, stay viable for a few more days, that can wait until the heart starts to reperfuse.
So can you measure the integration into the heart or the
Yeah, we can based on how the signal is integrating with the heart and also how the heart movement corresponds to the movement of the signal from the cells. Yep.
Where do the stem cells come from?
Generally it's from the patient themselves. So they take it from the iliac crest, and it's actually quite painful. They kind of drill into the iliac crest and suck out some bone marrow. There's work trying to find a universal donor. So if one person could do it and then you just grow up those cells, but just for safety reason, it tends to be from the patient themselves. So it's called an autologous donor. You donate to yourself. Yeah. Okay, well, you guys can go to the pool.
Okay, well, thanks so much. We really appreciate your contribution here, and it was, thanks.
Happy to do it.
Okay.